In the present study, we wanted to investigate whether we could reliably decode visual colors from EEG measurements. Multivariate methods have been used to decode a variety of visual dimensions. However, studies have shown that eye movements are a strong confound if you try to decode any spatially defined features such as location, spatial frequency, or orientation. So here we wanted to investigate how well we can decode the color dimension. The present data was taken from a working memory experiment where 30 participants were presented with 2 gratings, each with a color and an orientation, and our aim was to decode all 4 features at the visual presentation phase. So out of all 48 different colors and orientations, we extracted 12 different bins. And first of all, we checked if there is any univariate difference between 12 different colors. But there was no difference between any of the colors or the orientations. So perhaps color information can be decoded using the pattern of activity across sensors rather than the univariate signal. So if this is the case, we would expect that after we present a pink stimulus, that the classifier would predict there was most evidence for pink or red colors and not for green or blue colors. And we would expect to see a pattern that looked something like this. And we will do this for all different colors, and then we'll put the target color at the center of this y-axis. And then we're going to take a cross-section of every single time point, and we will convolve this with a cosine. Now we will obtain a single value for each time point, as illustrated here. Okay, so now let's have a look at the data. And yes, there seemed to be a very reliable ability to decode color from EEG measurements, as well as orientation. Although this does not provide much information where in the brain the signal is encoded. So to extend our results, we want to see that where out of these 61 electrodes we could most strongly decode color. So we ran a searchlight analysis, taking small groups of electrodes, and we did this for every single feature. So we found that posterior electrodes, contralateral to the presented stimulus, contributed most strongly. Now, it could also be the case that decoding is driven by just 1 or 2 colors. So to check this, we looked at the decoding evidence for each color between 150 and 350 milliseconds. So we are able to decode almost all of the colors reliably, and a large range of orientations as well. So next up, we analyzed the representation of similarity of all colors based on our EEG recordings. So we first composed a dissimilarity matrix by taking a color and then plotting which color the classifier guessed that you presented. And so we did this for every single color. And of course, we inverted all these values because we're looking at a dissimilarity matrix, not at a similarity matrix. And then we ran multidimensional scaling, and we observed a circular representation for both color and somewhat of a circular representation for orientation as well. So finally, we also tested the reliability of color decoding as a function of trial count and the number of study participants. So if you're planning to decode color in your experiment in some way, shape, or form, this might be a useful guide. So therefore we conclude that we're able to decode parametric color with a strong contribution of visual electrodes, and this is likely to reflect perceived color rather than the verbal labels. So thank you for your attention and for stopping by the poster. And if you have any questions, I'll be more than happy to answer them via the chat or via email.